Thank you very much, Maria Teresa. So we'll move to the next speaker, who will be Alfredo Miñano from Rodríguez Álvarez Group at the Autonomous University at Barcelona. So if you can upload your presentation, Alfredo, and go ahead. Thank you.
Good morning, everyone. I would like to thank the Foro organizer for giving me the opportunity to share our latest results about the AMPA receptor endocytosis accompanied by ACAP degradation. NMDA receptor-dependent long-term potentiation and long-term depression in excitatory hippocampal synapses are believed to play a fundamental role in learning and memory. Several reports have shown that changes in synaptic AMPA receptor number, properties, or subunit composition are involved in LTP and LTD. Those changes in AMPA receptors involve many actors responsible of the regulation of the trafficking, being phosphorylation by pKa and the phosphorylation by calcineurin key events. Although there is increasing evidence that A-beta oligomers might trigger signaling cascades similar to LTD, the actual mechanism at the postsynaptic membrane affecting AMPA receptor function at early stages of the pathology remain under discussion. Here emerged the role of a more than scaffold protein, the A kinase anchoring protein, 79 in humans and 150 in rodents. ACAP is a postsynaptic scaffold protein that anchors pKa, pKc, and calcineurin to the PSD, regulating AMPA receptor phosphorylation, trafficking, and activity associated with LTP and LTD. ACAP is linked to both AMPA and NMDA receptors by additional binding to the MAGUC scaffold proteins. Targeting ACAP to dendritic spines in an activity-dependent manner regulates trafficking, spine enlargement, and synaptic function. Thus, antagonistic regulation or the regulation of pKa and calcineurin activities together with the cycle of ACAP in and out of the synapses are major molecular mechanisms controlling synaptic AMPA receptors. However, it is still unknown whether degradation of ACAP is triggered by LTD or oligomers and whether it could be associated to AMPA receptor endocytosis. Analyzing the protein levels of AD brain human samples, we observe a significant correlation between ACAP and GLUA1. And this data caught our attention, and we decided to explore what happened to ACAP under those circumstances. Firstly, we want to observe whether our primary hippocampal neurons present the characteristic features after chemical LTD treatment, sharing 845 GLUA1 dephosphorylation and a decrease of GLUA1 AMPA receptor levels. Western blot analysis revealed a time-dependent decrease of serum phosphorylation after chemical LTD induction. Moreover, significant time-dependent changes were also found in GLUA1 total levels after an NMDA stimulus. A time-dependent reduction in surface GLUA1 containing AMPA receptors was also observed by biotinylation and Western blot analysis. Previous reports showed that chemical LTD causes a redistribution of the scaffolding protein ACAP away from synapses. However, it has not been reported whether this redistribution is also associated to changes in total ACAP levels. So our results indicate that chemical LTD significantly reduced ACAP levels in a time-dependent manner, as shown in this immunoblot analysis. After our finding that chemical LTD produced a decrease in ACAP, we wonder whether changes in ACAP go beyond synapse relocation and whether its degradation was mediated by proteasome activation. We assessed ACAP levels by confocal microscopy, retreating neurons with a specific inhibitor of the proteasome activity, MG132. We observed a significant reduction in ACAP immunoreactivity at dendrites after chemical LTD that was blocked by MG application. Moreover, we analyzed ACAP levels at PSD95 clusters, and we observed that MG were able to maintain ACAP at the synapse after chemical LTD. Our data suggests that not only the redistribution of ACAP, but also its degradation may play a role in the early stages of LTD. To determine whether chemical LTD-mediated ACAP decrease require ACAP ubiquitination, we check ubiquitination levels after chemical LTD induction in the presence of MJ in culture neurons. Accordingly, while chemical LTD reduced ACAP levels, pull-down results show a significant increase in total ubiquitin-conjugated ACAP observed in ACAP shift when overexposed. The results suggest that synaptic ACAP degradation by ubiquitin proteasome system could be an important factor in chemical LTD-induced synapse regulation. Synaptic distribution of ACAP is a key factor for synaptic expression of AMP receptors. So previous reports have shown that removal of ACAP from synapses toward the cytosol in dendritic shaft or soma elicits synaptic AMP receptor endocytosis. In this context, we explore ACAP silencing effect on AMP receptors. Using shRNA-mediated acute knockdown of endogenous ACAP, we were able to effectively reduce ACAP expression in cultured neurons. Interestingly, silencing decreased serine phosphorylation and GLUI1 total levels. Consistent with the results observed in immunoblotting, ACAP knockdown reduced both synaptic and dendritic GLUI1 levels, followed by a decrease in surface GLUI1 expression. These results show that a decrease in ACAP endogenous levels, either by chemical LTD induction or shRNA-mediated knockdown, triggers GLUI1 containing AMP receptor endocytosis. Therefore, we studied whether overexpression of ACAP was able to rescue AMP receptors endocytosis produced after chemical LTD. According to previous studies showing that ACAP overexpression somehow increases basal AMP receptor surface expression, neurons overexpressing ACAP showed a slight, not significant increase in serine phosphorylation without affecting GLUI1 total levels. However, ACAP overexpression did elevate surface GLUI1 leveling compared with basal. So we observed that when chemical LTD was induced in ACAP overexpressing neurons, surface GLUI1 levels remained unchanged. So once we observed this relationship between ACAP and GLUI1 on chemical LTD, we wondered whether ACAP was also affected by oligomers, not only at short exposure, but also chronically, becoming a key protein in early Alzheimer's disease. Here we advanced some of our unpublished data showing that oligomers induce ACAP degradation and GLUI1 containing AMP receptor endocytosis by the same mechanism than LTD at short term. Moreover, we have observed that prolonged absence of ACAP levels affects GLUI1 not only at short term, but also at long term, affecting its presence in the synapse by affecting protein expression. Analysis of the GLUI1 promoter activity using luciferase reporter show us that both knocking down ACAP and the presence of oligomers affect GLUI1 gene expression. So we are working on deciphering the mechanisms by which the absence of ACAP could be deregulating not only GLUI1 containing AMP receptors trafficking, but also gene expressions, leading synapse dysfunction at early stages of Alzheimer's disease. So our take-home message from our recent work is that ACAP could be pivotal for synaptic AMP receptor regulation, emerging as a key protein in early synapse dysfunction in Alzheimer's disease. So finally, we would like to acknowledge lab members, especially those involved more in depth with this work, or collaborators and the institutions for funding. And thank you very much for your attention.